Hello, Thumbi. Hi, Thumbi. How are you? Welcome everyone to the community dialogue about HIV cure research. We are incredibly fortunate today. In fact, let me just quickly get this live. Quickly make sure that this is going to go live. Okay, well, while we go ahead and wait for that to go live, great, we can go ahead and start sharing. Great, so we're really fortunate today to have a wonderful community dialogue about HIV cure research with a wonderful speaker, Professor Thumbi Ndugu from the University of KwaZulu-Natal. Not only did he give a fantastic plenary yesterday at AIDS 2020, Dr. Ndugu is the Deputy Director and the Max Planck Research Group Leader at the Africa Health Research Institute in Durban, South Africa. He's a professor and Victor Dias Chair in HIV/TB research at the Nelson Mandela School of Medicine, the University of KwaZulu-Natal. He holds the South African Research Chair in System Biology of HIV/AIDS, and he is an adjunct professor of immunology and infectious diseases at the Harvard Chan School of Public Health. He's the program director of the Sub-Saharan African Network for TB and HIV Research Excellence, and is a research capacity building initiative funded by the African Academy of Sciences and the Wellcome Trust. I could really go on and on about Thumbi Ndugu. He's not only just a leading researcher in the field, is just a really wonderful person to be around, and is been thinking about cure research, and particularly how to bring cure research to resource limited settings for quite some time. And his work is really culminating now in a clinical trial that he may be able to chat with us about. But for those who need a background on this session, unfortunately, AIDS 2020, given the state of the world, has gone virtual, and so we can't all be together in person. But what it has allowed us is a chance to really bring the global village truly global, and allow us to have these types of sessions and reach a much larger audience than we would have had we been in San Francisco together. So this is part of our pop-up chat series as part of the Research Literacy Networking Zone, which is a space in the Global Village. And the Global Village, for those who don't attend the AIDS 2020 conferences or AIDS conferences in the past, is a space that is open to the public, and it's really about sharing knowledge. And our goal of this zone is really about targeting that knowledge and allowing advocates and community members to learn more about the research that's happening in the fields and how it can translate into their life. So we are incredibly fortunate to have Dr. Ndugu today, and we'll just be having a Q&A. So just really having a dialogue with one another. So I have some questions to start off, Thumbi, but we do really encourage everyone to use the chat feature and to ask questions here. I'll also be taking questions on the YouTube space as well. So apologies if you see me toggle a little bit between my phone and the computer screen. I'm just trying to make sure we flag questions from everywhere. So first question, welcome Thumbi, how are you?
Thank you very much, Jessica. It's a pleasure to participate in this forum.
Great. So the first question we have is really, what are we talking about when we talk about cure? I think we all have a little bit of a different meaning, but what do researchers mean when they say cure?
Okay, that's a great question, Jess. And I think there are two kind of widely accepted approaches to HIV cure. One would be where we achieve a complete eradication of the virus from the body. So a cure strategy where basically you get rid of the virus completely from the body. That's one strategy. And the other strategy would be what we call remission. In the case of remission, this is where somebody still has HIV in their body, but the virus is present at such low levels that it is not causing disease. And also the virus cannot be transmitted to another person. And this is all in the absence of antiretroviral therapy. So the person is not taking antiretroviral therapy, but the virus is still present, can be detected, but it is at such low levels that the virus is not causing any harm to the individual, and also the virus cannot be transmitted to another person. So if we can achieve that remission, so the person is not on antiretroviral therapy, then that would be the second strategy. And that is thought to be the most likely possibility for most HIV-infected people.
Great. So when you say is the most likely, is remission a step to cure?
I think you could see it as a step to cure. Yes, I think you could. It could be a step towards completely eradicating the virus, but it could also be an end in itself. I think the problem with a chronic disease such as HIV is the fact that it is very difficult to prove that somebody has actually been completely cured of the virus. It's almost the same kind of scenario that we are faced with when we talk about somebody with cancer, for example, where it is very difficult to prove that the person has been completely cured. So we prefer to use the term remission because it is possible that there is still, for example, when somebody has undergone cancer treatment and it is declared cancer-free, it is still possible that there is some cancerous cells that are lacking somewhere in the body, and we just can't detect them. So we prefer to actually use the term remission because it's very difficult to prove a cure. And so I would see remission as a step, certainly a very positive step, because the person can live without antiretroviral therapy, and the virus is not causing any harm, and the virus cannot be transmitted to somebody else. It is present at very low levels. But it can also be an end in itself, in the sense that if somebody can be on long-term remission for 20, 30 years, where they no longer need antiretroviral therapy for the rest of their life, even if you can detect small amount of the virus in the body, that is also quite acceptable, I think, in my opinion. But of course, we all would like to have the scenario where the person can completely eradicate the virus.
Yeah, no, absolutely. And one of the things you talked about in the plenary yesterday, I think we're using a lot of terminology here. So if you don't understand a word that we're saying, please raise that in the chat and in the comment section on YouTube, so that we can make sure we clarify what we're saying. But we talked about the reservoir. And one of the questions to me that came in was, is the reservoir resistant to ART? Is that what makes it so hard to cure HIV?
Okay, Jess, let's first, I think, define that word reservoir, because we use it a lot in the scientific arena, and I think it confuses some people. What do we actually mean by a reservoir? So I think in simple terms, what we are talking about is when somebody is on antiretroviral therapy. We know that when you are on antiretroviral therapy, your viral load, which is the amount of virus that is in free circulation in the blood, in the body, is undetectable. it should go to undetectable within a few months of starting antiretroviral therapy. The problem is that it does not mean that you have completely eliminated HIV from the body, because we know that there are small amounts of the virus that are still hiding inside cells in the body, in the blood, or even in tissues, such as, for example, in the lymph node, in the central nervous system, in the brain, in the gut. There are some viruses that are still hiding in cells, inside the cells. So that virus that is hiding in the cells is not a free virus. It's not, you cannot detect it if you do a viral load assay. And that's why we say the viral load is undetectable. But we know that there's virus, small amounts of virus that are hiding in certain cells in the body. Now, that virus that is hiding in the cells in the body is what we call the reservoir. And that is what is very difficult to eliminate, because HIV antiretroviral therapy does not get rid of the reservoir. It cannot completely eradicate the reservoir. And the reason for that is because HIV, the antiretroviral drugs that we use, what they actually do is they prevent the virus from replicating. They prevent the virus from making copies of itself. But the antiretroviral therapy does not completely eliminate those cells that may be infected and are present in small levels in the body, but they are not actively producing virus. So that's what we call the reservoir, that virus that is hiding in the body. And that virus, in fact, the reservoir is in fact resistant to antiretroviral therapy. It cannot be eliminated by antiretroviral therapy.
Mm. So that's a really great point. And I think that resistance gets lost sometimes, because one of the follow-up questions too was, when that virus comes out, would ART stop it, or does it actually infect other cells again?
Okay, so if somebody is on antiretroviral therapy, we have already explained that the free virus is completely eliminated. It goes to undetectable levels. But the virus that is hiding in the cells is still present. And if the person stops taking antiretroviral therapy, what then happens is that the virus comes out and the virus starts replicating. So now, if you then reintroduce antiretroviral therapy, let's say the person stops antiretroviral therapy, the virus will come out from hiding and it will start replicating. But that virus can actually, in most cases, that virus can be inhibited again by antiretroviral therapy. So if the person goes back on antiretroviral therapy, that virus will be inhibited and you will not have free virus. But remember that again, there will still be small amounts of the virus will again go into hiding in the cells. So it becomes almost like a cat and mouse game, in the sense that if you stop antiretroviral therapy, the virus will come out of hiding, but the virus will not be eliminated by antiretroviral therapy, because as soon as you stop antiretroviral therapy, the virus, as soon as you start antiretroviral therapy, the virus goes back into hiding. So antiretroviral therapy in itself can never completely eliminate the reservoir.
Yeah. So I think that's such a helpful explanation, this idea that the virus, right, when it hides, it's not immune to, or it is immune to ART, mostly, right, because it hides in the body in the same way that ART doesn't impact all immune cells. But then when it's free virus, when it's floating around, that's when ART can do its job. And so it can actually be impacted by having antiretroviral therapy, but some virus still goes back into hiding. And so you get this, you get this circular game. Now, you used a couple of words in there: assay, viral load, both of which, as we know, are tests to measure the amount of virus. So something that comes up a lot is when we monitor HIV cure, cure and research, how do people measure the reservoir? What kinds of measurements are they taking? Is it antibodies? Is it viral load? What are the things people are looking for?
Again, that's a great question. Again, Jess, and actually a difficult question. So generally speaking, most people will have heard of the viral load assay, because that's the assay that we commonly use to monitor people who are HIV infected and to see whether antiretroviral drugs are working. So the viral load assay generally measures the amount of free virus that is floating freely in the blood, and that tells you whether the antiretroviral therapies are working or not. However, when you talk about the reservoir, we are now measuring the virus that is hiding inside the cells. So it's completely different from the viral load assay. And there are various ways to measure that reservoir. One way is to just measure the amount of cells that actually contain some piece of HIV DNA. So you can quantify the amount of cells within the blood that contain HIV, and then you can say that that's a reservoir. Now, the problem with that kind of way of measuring the reservoir is that we also know that some of that virus that is hiding within the cells is actually not productive virus. In other words, some of the cells will be infected with defective HIV that cannot replicate. So sometimes when you measure the reservoir just by measuring the amount of HIV DNA within the cells, you may overestimate the amount of the reservoir, because not all the cells are, in fact, even though some contain HIV, not all of them are capable of producing infectious, what we call infectious virus. Mm-hmm. So scientists have also developed other laboratory assays where they can measure the amounts of cells that are able to produce active or replicating infectious virus. So in other words, what I'm saying is that the measurement of the reservoir itself is not a straightforward thing. It's a very complicated process, and there are various assays, some of them just based on measuring the amounts of cells that have HIV DNA. Some are based on measuring the amounts of cells that can actively produce infectious virus. And there are even other assays that look for virus hiding within tissues. So there are very many different ways to measure the reservoir. And this is one of the most active areas of HIV research, trying to understand what is the reservoir, where is the reservoir, because all of this has implications for how we can get rid of the virus and eliminate it from the body.
Great. I think, measuring the reservoir is really challenging, and I imagine that is what makes clinical trials for cure challenging as well, trying to decipher between those different tests. But I want to take a step back before we kind of dive into that a little bit deeper and think about what are the ways that people are trying to cure HIV? What are the strategies being used or being thought about in the research community?
Okay, there are various strategies for curing HIV, and I think I will mention. I'll mention 5 of them. And each one of them is kind of technically complicated, so I'm not going to go into all the details, but generally speaking, there are about 5 strategies. One is what we call block and lock. And block and lock, what it means is that you identify the HIV infected cells that are hiding in the body, the cells that contain HIV, and you do something to those cells so that they are no longer able to produce virus. In other words, you lock that virus within the cells and inactivate those cells or the virus so that those infected cells that harbor the virus are no longer able to produce infectious virus. So basically, you're trying to contain that virus within the infected cells. So that's the block and lock. You're basically trying to lock that virus inside the cells so that it can never come out. That's one strategy. Another strategy, which is actually much more popular in terms of what people have tried to do, is what we call kick and kill. In the kick and kill strategy, what you do is you identify the cells that have HIV, that have the reservoir, that are latently infected with HIV, and you somehow activate the cells, those cells, so that they start producing the virus. And if you activate those cells, the cells will die. And you can then get rid of those cells as well. So you basically kick those cells so that they can start producing the virus. They produce the virus, and then you kill those cells that are producing the virus. So that's another strategy. The third strategy is what we call immunotherapy. In immunotherapy, you activate the individual's immune system so that they can start to see the virus that is, or the cells that are latently infected, the cells that are containing the virus. And then you use the immune system to eliminate those cells. And another strategy, the fourth one, is what we call gene editing. So you can actually change the cells themselves so that they become resistant to HIV infection. So for example, we know that for HIV to enter cells, you need certain receptors. Certain specific proteins are needed on the cell surface for HIV to enter the cell. So you can eliminate some of those proteins that are needed for the HIV to enter cells. So that's what we call gene editing. And that has actually been shown to work very well. For example, for the Berlin patient and the London patient, this is actually what happened, is that those persons received a bone marrow transplant, and they received cells that were resistant to HIV, so they could no longer be infected with HIV. So gene editing is another approach, but it is very expensive and it is very risky clinically. So it's not something that a lot of people at the moment are pursuing, even though there is some hope that in future these kinds of therapies could be useful. But at the moment, they're not going to be widely used. And then the fifth one that I would like to discuss, Jess, is a combination of those approaches. So most scientists believe that that no single approach is actually going to work very well. And actually what we probably need is a combination of one or two or three approaches to actually get rid of the virus.
Yeah, and just to kind of recap, to make sure we're all on the same page, and because that was such a great explanation, block and lock, that is thinking really towards remission, that remission approach, right? So it's not trying to eradicate the virus, it's trying to freeze it in place.
That's correct.
Yeah, kick and kill is eradication and thinking about getting that virus out. So waking it up and then figuring out how to clear it from the body. So getting it out of the reservoir and clearing it. The immune-based strategies, like you mentioned, is thinking about enhancing that. That might be, like you said, in a combination, might be combined with that might be the kill portion of a kick and kill approach. Yes. And then finally, gene editing, which we've seen work, right? We have the proof of concept about from both the Tim Brown, the Berlin patient, and the London patient as well. So So that is sort of a different approach, but like you said, this idea of a combination, bringing them all together, which, you know, is very, it's obviously like antiretroviral therapy, right? You need sort of all of the tools in your toolbox to make sure that you're getting the virus out.
Yes.
And on that, you know, there has been news, obviously, yesterday at the conference of a potential third cure that didn't involve gene editing. And so one of the questions that has come in about what is your perspective or your take on the Sao Paulo, I guess that's what the name is, the Sao Paulo cure, the potential cure from Brazil.
Okay, so I have to admit that I'm not familiar just with the Sao Paulo patient.
So it's so I'll just say it's very new data. And Steve Deeks, who's another researcher in the field, specifically is quoted saying that this is not a cure, but it is an interesting case to study. It seems like the individual actually got treatment intensification. So they were given the standard triple drug cocktail and then also had Maraviroc and I believe dolutegravir. Yeah. And then a vitamin B3 added to their regimen as well. And then went on an ATI and has seemed to be continued to be or off therapy.
Actually, maybe I can comment that in order for all these cure strategies to work, it seems that one of the most important thing is to reduce the reservoir, the size of the reservoir, the number of cells that are infected, that are in the body, that are hiding in the body. And one of the most effective ways to do that is actually through antiretroviral therapy, particularly antiretroviral therapy that is given very early on during HIV infection. So the earlier somebody starts antiretroviral therapy, it seems that the lower the amount of virus that is hiding in the body. So antiretroviral, particularly early antiretroviral therapy or intensified antiretroviral therapy is one way to actually reduce the reservoir and also reduce a lot of immune dysfunction. And that allows these other strategies, such as the immunotherapy, to come into play. It makes it much easier. The bar is lower for those strategies if somebody has a reduced amount of virus that is hiding in the body. So it seems like intensification of antiretroviral therapy or giving antiretroviral therapy very early during the during HIV infection is a very good way to prepare somebody for a cure strategy, no matter what the cure strategy is. And I would imagine that in that Sao Paulo case, this is what happened, is that the individual probably received antiretroviral therapy or intensified antiretroviral therapy early on during their infection, and then the researchers come in with immune-based therapies, and there are many of them. They could be vaccines, they could be antibodies, they could be other approaches. And this kind of combination approach is one of the most promising cure strategies. And I would like to add just that this is probably, I'm excited about this because it's one of the strategies that is likely to work in resource limited settings where most of HIV infection is. So if we can treat people early and then come in with the immune-based therapies such as monoclonal antibodies or vaccines or other immune modulators, this might be a way for us to actually achieve long-term remission, even in resource-limited settings.
Yeah, and I do want to get into that piece now as well, because yesterday in your plenary, you talked about the early, some of the early treatment, and I know it was towards the end, but you were talking about a study that you are going to be part of as well in Durban, South Africa. So I was wondering if you could talk a little bit about how early treatment is playing a role in leading up to that clinical trial, and maybe a little bit about the clinical trial itself.
Thanks, Jess. This is a study that we are very excited about. I have to emphasize it's a research study, so when you do a research study, you don't know whether the concept is really going to work or not. Yeah. But we are excited about it because of some of the reasons that I've mentioned, which is, first of all, what we do, because of the fact that we have high rates of HIV infection here in KwaZulu-Natal, in South Africa, we identify women during the early stages of HIV infection, what we call the acute phase of infection, very early. And then we give them antiretroviral therapy immediately, within a day of us detecting the virus in their blood. So, in other words, we are introducing antiretroviral therapy very, very early, as humanly possible, as soon as we can detect that this person got infected. And as I mentioned, early antiretroviral therapy, we think reduces the reservoir, the size of the reservoir, but it also helps to reduce the virus diversification. So the virus doesn't become very diversified. And this then sets, what we have also shown actually in this study is that when you introduce antiretroviral therapy so early, you preserve the person's immune system. So one of the hallmarks of HIV infection is that it destroys the immune system, particularly CD4 T cells. Now, what we have shown is that if you introduce early antiretroviral therapy, you actually preserve CD4 T cell counts, and those people who are treated so early, their CD4 counts look almost exactly like those of people who are not HIV infected. So these people are not yet cured of HIV, but the reservoir is low and they have very good immune function. So we think that we may be able to then intervene with an immunotherapy, such as a broadly neutralizing monoclonal antibody. And a monoclonal antibody is a protein that can bind to HIV and inactivate HIV and prevent it from entering cells. And we have actually shown that some of the monoclonal antibodies that we have tested in our setting are able to inhibit some of these viruses that infected some of these individuals in our cohort. So these are the kinds of strategies that we think that with early treatment and then immunomodulation with antibodies and maybe a combination of other immune-based therapies, that actually we might be able to see long-term remission in some of these individuals. So this is a concept. It's a research idea. We are going to be testing it possibly next year, and we are excited that this could be a proof of concept type of study that shows whether this kind of strategy can work in terms of curing people of HIV, or at least leading to long-term remission.
So Thumbi, there's a lot of questions now coming in the chat feature, and Moses Supercharger, who is a fantastic advocate from Uganda, actually has a question. So I'm going to begin to unmute some folks, but I think it's worth just mentioning as we get some folks unmuted, is that this cohort, this research study that you're talking about, is actually a prevention cohort that has been put in place that yes, identifies people who have been infected with the virus very early, but also is providing a job program and a real prevention program for these young women in the community. So so cure and prevention are on the same spectrum.
Yeah, that's a really great point actually to emphasize, because what we are really trying to do first and foremost is that we know that we have high rates of HIV infection. And so we asked ourselves, what could we do to try and prevent HIV infection, particularly among young women in a resource poor setting? And we thought that one way to try and prevent HIV infection is to empower people by preparing them with skills so that they can find a job and enter the workplace and be prepared to enter the workplace. And so we set up a program where we teach people, young women, how to use computers. We also give them other sort of life skills. We show them how to, or we teach them about HIV prevention and empowerment of women. And they come to our clinic for these classes, empowerment classes, twice a week. Now, when they come into the clinic, we also take a finger prick blood draw to test for HIV, because these are obviously people who are also going about their normal lives. So despite our messages of HIV prevention, we know that there is also some risk behavior going on. And what we have been able to do with that program is, in fact, to empower women. We have had about 80% of them actually entering the job market. But unfortunately, because of the fact that we have high HIV infection rates, we have also had almost about, since we started the program in 2012, we have had about 100 of the women getting HIV infected. So this is it's a great program in terms of empowering women, also for testing HIV prevention strategies, including PrEP, which we offer. But in the instances where people also get HIV infected, in the unfortunate circumstances that somebody gets HIV infected, we also offer them immediate antiretroviral therapy, and we hope that we can then intervene with a cure strategy down the line.
Yeah, so I think that that's just a really important message, because I do know that some of the questions that have come in over the various forums are how can advocates help and what can advocates do? And I think that this is a really model program that says prevention first, but if infection happens, you know, cure research is possible and can really go hand in hand. But Supercharger, I've gone ahead and unmuted your line. I don't know if you want to ask your question directly. Supercharger. Supercharger, can you go ahead and ask your question directly?
Yes, the quick one. I have like 3 questions. One, I wanted to know whether kick and kill is the same as shock and kill. And also the doctor said that when they kick and kill the virus, they're trying to identify cells and activate them to produce more cells. That is a little bit confusing. Why activate them to produce more cells? And why don't you just activate those cells to come back so that ARVs can kill them? And the last one is about the immunotherapy. There is not so much enthusiasm and energy around research to do with immunotherapy when it comes to curing HIV. There is a lot of investment and enthusiasm into complicated things, the block and lock, the gene therapy. And I think as a community member, immuno-based therapy would be something easy to do, but the investments into it, I'm not seeing them as a community member. Thank you.
Thank you very much, Supercharger. It's actually a pleasure to meet online again. So kick and kill and shock and kill are really the same thing. Those are just different terms that people use, but really they're, for the most part, they're referring to exactly the same strategy. Okay. In terms of what I said, when you do kick and kill, what you're trying to do is to activate the cells that have HIV in them and are hiding in the body. Because the problem with these hiding cells is that they can hide in the body for many years. In fact, these cells can last in the body for 10 years or even 20 years and not produce virus. But somewhere down the line, they start producing virus, and we don't always understand why. they start all of a sudden producing virus. So what you are doing to do in a kick and kill strategy is you are activating those cells so that they can come out of hiding, basically, produce the virus, and then you kill those cells, and you kill the virus that is coming out of those cells with antiretroviral therapy. So you're not activating the cells to produce more cells. You are activating the cells so they can produce the virus, and so that you can also kill those cells that contain the virus, but also kill the virus that then comes out, so that you don't have ongoing infection. So I hope that is clear. And then your last question about immunotherapy, you're absolutely right that immune-based therapies might actually be the best approach at the moment, because of the fact that we have a lot of experience with immune-based therapies in the clinic. We know that vaccines, for example, are, for the most part, they are safe. We know how to use them. So for me, immune-based therapies, and they can also be used in resource-limited settings where most of HIV is. So I think that these immune-based therapies are the most promising ones, particularly for us in resource-limited settings. And I think that we need to do much more research to investigate whether these immune-based therapies can work in HIV cure, and while still pursuing the other strategies like gene therapies. Like I said, I think gene therapies are very expensive, and they are not probably ready for prime time in resource-limited settings. But immune-based therapies might actually be a way that will lead us to eventually achieving a cure, particularly in some of the most heavily burdened communities.
Great. So Thumbi, just to follow up on that, there's a couple of questions that have come up in the chat feature. And one is from Yvonne about passive immunization. So are there specific antibodies that you can use, and are they available in Africa, especially Kenya?
No. Okay, first of all, I think it's important for us to state right from the get-go that at the moment, there are no antibodies that can cure HIV or even stop HIV that are available in the clinics. So if somebody tells you that there are antibodies that are available that you can go and buy in a clinic or in a hospital somewhere, they would be lying to you, because everything that we are talking about is under investigation. It's research that is being done. So we have antibodies, yes, that have been shown in laboratories to inhibit HIV, but these antibodies have not been proven that they can actually work to cure anybody of HIV. There are some good ideas about how they might be used, but we need to do the studies to prove that in fact these antibodies do work, both for prevention, but also for cure strategies. And this is the work that is ongoing. And this is the work that I think as activists who are in this call, we should all be involved in, is to promote this work, is to say this, we should be doing this in the places with high HIV burden. But at the moment, these antibodies are, some have been tested in monkey studies. There are some that have been tested in human studies and have shown some promise, but none of them has been shown to be good enough for it to be licensed. So we need to do more studies to actually prove that these antibodies can work. And some of them, like I said, are showing a lot of promise in the preliminary studies, but we just need to generate much more data before we can tell anybody that they can use these antibodies in the clinic.
That's a really great point. And some of you on the call may know in the prevention side, there's a trial called antibody-mediated prevention, which is testing a broadly neutralizing antibody, which is what Thumbi is talking about, for prevention. And those results are due sometime this year. We should hear about the results of that large clinical trial. But again, that is not going for licensure. It is just a proof of concept study. So it is important that, yes, these are not available now, but there's a lot of exciting work. And Thumbi, just on that piece, yesterday in your plenary, you had a really great slide about the multiple different kinds of BNABs, or broadly neutralizing antibodies, that are available. And we're also discussing in the vaccine space, the actual and sort of immune prompting strategy of a vaccine. This idea of a mosaic, the idea that you may need different, that there are different strains, and so a vaccine has to cover all of them. And broadly neutralizing antibodies may need to do the same, right? That you may need a combination of broadly neutralizing antibodies to work on different viruses. And I'm wondering if you could talk a little bit about that idea of the different strains and what it might mean for HIV cure research. Is there going to be a one-size-fits-all cure? Do we need different cures for different populations?
Well, this is another great question. I think the problem we have with HIV, as we all know, is the biggest challenge of HIV is the genetic diversity of the virus. And when we say genetic diversity, what we mean is that HIV, even within one single individual, actually is not just a single strain, it's different strains of the virus that are circulating within the body, because the virus has the capability to mutate a lot, much more than other viruses. So in fact, if you compare HIV with the SARS-CoV-2 virus that is now causing the COVID-19 epidemic, I think that HIV is a much more challenging virus because of the fact that it mutates so much, which is not the case, for example, with the SARS-CoV-2 virus. So one of the challenges with HIV is whether we are going to be able to identify an antibody that can kill all the strains of HIV, or even a vaccine that can stop all the strains of HIV. And the likelihood is that no single antibody or no single vaccine immunogen is going to be able to do that. And so what scientists are doing is to think of combinations of antibodies, very much like we do for antiretroviral therapy. So when you get antiretroviral therapy for HIV, you get a combination of 3 drugs. And when you have a combination of 3 drugs, that forces the virus, that hits the virus at various sites so that the virus is unable to maneuver and get out of that situation. And therefore, then you can suppress it. The same thing is being thought about when it comes to antibodies. So we have broadly neutralizing antibodies that target different parts of the virus, and it is unlikely that just one single antibody is going to work. But it is also likely, just like in ARVs, that a combination of antibodies is going to work. So that is what researchers are going to be trying. And the same thing applies to vaccines. It's a combination of maybe vaccine immunogens that are targeting the virus at different sites, and that might then lead to a cure or effective prevention.
You know, I think that just highlights even more why we need to have this research being done in resource-limited settings to help potentially discover more broadly neutralizing antibodies, but also figure out what combinations might work in different populations. So I know we only have 10 minutes left, and there's a host of questions flying in, but Sindra, I did just want to take a moment for you to ask your question. So can you go ahead?
Sure, thanks. Just really quickly, this is a follow-on, Thumbi. Thanks so much about the young women in KZN who have, I think it's been a few years now that they're, you're watching them for when they seroconvert, you get them on early treatment. And my question is, why it seems like you haven't started them yet with the immunotherapy, but why not use the antibodies that we have? Why have we not started those with these young women or any of the vaccines? why are we waiting? What are we waiting for?
Okay, that's a great question again. So at the moment, of course, when somebody gets HIV infected, we use the standard approved drugs for treatment, which is antiretroviral therapies. But we have now identified quite a number of these women, and we have been able to place them on antiretroviral therapy for up to 2 years and follow them up. And so the next step now is to see if we intervene with monoclonal antibodies, would that lead to long-term remission in these women? And that's exactly what we are planning to do. So we are in the process of acquiring the antibodies from the various manufacturers. We are in the process of getting approval for the study from the ethics boards as well as the regulatory authorities. And then we hope that we can start this study with these antibodies within the next 1 year or so to see whether in fact we can get remission. So that's what is being planned.
It just seems like it's taking a while, but I guess there's, right?
It does take a while because we must get all the ethics approvals and the regulatory approvals. Remember that some of these antibodies, sometimes you experience problems with the manufacturing them, particularly for human use. Because even though sometimes they are very effective in the laboratory, you still have to manufacture them for human use and certify that they are safe to be used in humans. So all that process is a fairly complicated process. And the other thing is that also we want to test the viruses from these individuals to make sure that the antibody, that the virus that they have is actually sensitive to the monoclonal antibodies. So all of that is ongoing work in preparation for the clinical trial.
Okay, thank you.
These are really great questions. And
I have a quick one for Jessica. Do we still have some time?
Yes.
Jessica, I have a question to you and a quick one also to Dr. Ndungu. The one for you is about the pharmaceutical companies, really, in relation to the HIV cure research. I mean, there is no enthusiasm, there is no energy around pharmaceutical industries. And just last week, I realized that actually as community, we don't even know the kind of investment they are doing into HIV cure research. And it disturbs me as a community member. And the last one to Dr. Ndungu is about the problem of mutation. This is the question I've asked researchers for the last 10 years. Where is the cure? And every day they tell me the problem is that the virus mutates. The virus mutates. What are you doing as researchers really to understand why this virus mutates the way it is and not other viruses?
So Thumbi, I can start with a pharmaceutical place question. I do think it's a really important thing that we do hear rumors a lot that people aren't investing in cure research, and that we know is not true. There is actually an investment in this area of research, and it's actually been growing over the last few years. So AVAC and the International AIDS Society do publish a resource tracking report, which we can make available to everybody on this call, where you can actually see the investment going up in this field of studying HIV cure research, not just from the NIH, but also from pharmaceutical and small biotechnology companies as well. In fact, the NIH has 6 collaboratories that collaboratories are sort of a collection of research organizations that are being put together with an industry partner whose sole focus has been to try to find a cure for HIV and learn some of the basic science that can lead there. That program has been ongoing now for 7 years, I believe, and is currently in the renewal phase as well. So the program is continuing on. And something, just to think about too, Gilead actually came out with a great new, it came out with some additional information on a new product a new type of drug called the capsid inhibitor. We won't go into too much detail, but it is something that could be a potentially wonderful thing for the cure field, like Thumbi said, in getting art on board and suppressing that viral reservoir very low, very early. So something to look for and to just keep your eye out for this week as well. But it is just the short answer is to say that pharmaceutical companies are investing in this and are engaged in this space. But as always, advocates can and should push for more resources, because as we know, this isn't just you don't have to take away from one area of research to fund the other. This is not an if then, this is a both situation. We can fund prevention and treatment and cure at equal measures. So Thumbi, I don't know if you want to touch about the viral diversity very quickly.
Yeah, I mean, we know a lot about why there's virus diversity. It's the nature of the virus. It has what we call, it has to do with the life cycle and how the virus enters the cell. And as it enters the cell and makes copies of itself, it has the capability to mutate and cause errors, mistakes in its own replication, which results in some changes. But it turns out that this is a good property for the virus because it enables the virus to evade the immune system. So this is a challenge for vaccine and for cure and for treatment, but it is not an insurmountable challenge. As we have seen with antiretroviral therapy, through using combinational approaches, you can actually overcome this problem. And I think that in a similar fashion, we may be able to develop immunotherapies or antibodies, for example, that can overcome this problem. Just also to say very quickly that although these strategies sound, I see somebody has commented about the strategies are very complicated. It is true that they are complicated, but I think also it is also true that there are some very promising and exciting ideas out there, and we need to test them, and including testing them in children, because this may actually lead us to and in fact there may be some advantages that children have over adults in terms of maybe achieving a cure. But we can have further discussions some other point about that actually. Yeah,
I think that's a great piece to really wrap everything up on. And I apologize. I know we didn't get to all the questions here, but we will share ways that we can continue to stay engaged in this conversation. But I think it's important to remember, to even get to ARVs, that research at one point was in the bench, was in the laboratory, and it seemed complicated. But science is really a wonderful thing, and the innovation that's come forward in the last 30, 40 years of the epidemic is been great, and it has moved very far. And cure research can have that spark as well. So they sound very complicated, but what has taken into a broadly neutralizing antibody for prevention is in phase 3 clinical trials in humans now for prevention. And so there is a lot of hope in this field and a lot of excitement and a lot of really smart, brilliant minds, Thumbi's included, working on these problems. So we certainly have reasons to be hopeful. So I know we're at the top of the hour and we all have busy schedules, so I do want to be a good steward of time. And I just want to thank you so much, Thumbi, for your time and your expertise and your brilliant way that you've really explained and broken down the research for us. It has been incredibly helpful to me, and I'm sure helpful to others as well.
Well, it's my pleasure, and really delighted to engage with everyone. And we are all in this together, and in fighting HIV/AIDS, and together we can achieve a lot. So let's keep up the fight.
Great. And for those, we will follow up, but you can continue this conversation, as I mentioned, on Engage, and we'll share resources on how to get there and some of the resources and questions from this call today. So thank you, everyone, and have a wonderful afternoon all.
Thank you.
Thank you.